Okay, this is the second lecture for the nervous system. This one is going to be some of the cellular structures and functions of the nervous system. So we'll talk about action potentials, we'll talk about neurotransmitters. This first picture is just a general structure of a neuron. Notice that we've got the dendrites, which are going to be the structures or the extensions of the cell membrane that receive a nerve impulse. There's little red arrows here. The nerve impulse is coming in from another cell, and then the axon, the blue arrow here, the signal travels down the axon away from the cell body. The axon is often myelinated. That's what we see here with these blue wrappings, and we'll talk about that a little bit more later. And then the axon ends with these little axon terminals, sometimes called terminal buttons. This is going to be the little rounded ends of the axon as it sort of branches out at the end. A nerve is a bundle of neurons. Hopefully this picture looks familiar or similar to the muscle structure we looked at last chapter. Notice that, again, we just have long, thin things bundled together into other cylindrical things. So if we start here at the small end, we've got our axon. This would be the axon of a single neuron. In this case, it's covered with myelin. And then we have our neurons bundled together to make a fascicle. The fascicles bundled together make a nerve. The nerve also includes blood vessels. So notice here that it's lots of neuron cells bundled together with layers of connective tissue and blood vessels. All right, in the muscle, we had epimysium, perimysium, endomysium. We're going to use really similar terms for this structure, except our second half of the word is neurium, but the same locations kind of apply. So try to number 1 through 6 here with these terms. You can pause the video if you want to. I'm going to go on and show you the answers in just a second. The epineurium is the big outer layer, so kind of think epidermis. Epi means around or outside. The perineurium surrounds a fascicle, so that would be number 2. The endoneurium surrounds a single neuron. And then number 4 here is the neuron itself. We've got the myelin sheath that coats the neuron, and we've got the blood vessels. I didn't get very creative with the rearranging of the terms. They're already in order if you didn't notice that already. So the function of myelin, we've seen myelin sheaths coating the axons, and you've probably read that myelin acts as an insulator. What that means is not that it really necessarily protects the axon or keeps it warm, but just that it helps to speed up the transmission of impulses. So here is kind of a chain of neurons. We can see the axon terminal of this first neuron, and it is in contact with the dendrite of the second neuron. This is the region where they sort of meet is called a synapse. They don't physically touch. There's a little space in between. And then once that signal is received by the dendrite of this neuron, it's going to be transmitted down the axon to the dendrite of the next neuron. And again, we'll have a synapse in between. The myelin sheath is kind of interesting. We've got some glial cells, which are sort of helper cells for the neurons, and 2 different types, oligodendrocytes and Schwann cells. Oligodendrocytes are the ones we're going to find in the central nervous system, Schwann are in the peripheral. And basically, they both create the myelin sheath by extending their cell membranes out and wrapping around the axon. So the red dot here is a cross section of an axon, and the myelin sheath is essentially a fatty coating that surrounds the axon. Here's another look at the myelin sheath. The purple outline here is all cell membrane of a Schwann cell. And then notice that we have these little spaces in between. So between one Schwann cell and the next, there are nodes of Ranvier, where the axon is actually exposed. Neurons send signals. So neurons are specialized cells that are able to transmit impulses. And keep in mind the direction of the signal. It's always going to go away from the cell body down the axon. And what actually happens when this is happening, when the impulse is being sent, is that the axon is depolarizing. And so we'll talk about what that means in just a second. But keep in mind that an action potential is the transmission of a signal, which is really just the axon depolarizing farther and farther down until it gets depolarized all the way to the end. So depolarization, polarization, let's just talk about those terms quickly. A neuron at rest is polarized, and all that means is that there's a charge difference across the membrane. In the case of the neuron, it's more negatively charged inside than it is outside. The reason for this is just that we have a difference in ion concentrations. We have these channels in the membrane that, when they're closed, help to maintain the separation of ions across the membrane. Notice that we've got more sodium outside the cell. These are the little blue dots. More potassium inside. You can think of a salty banana if this analogy helps you. Bananas have a lot of potassium, and if you have a salty banana, the salt is on the outside. Okay, so the action potential is going to travel down the axon, and it's going to happen just at the nodes of Ranvier, and then that action potential will spread. So once the node depolarizes, the depolarization will spread all the way till the next node. We'll talk more about what that means, but just notice that the myelin sheath is blocking the axon. The places where the depolarization is happening is at the nodes. The depolarization refers to the fact that the charges across the cell membrane are going to be evening out. So before we said that polarization is a separation of charge. Depolarization is when the separation of charge becomes less, when the charge inside becomes more equal to the charge outside. The way this happens is that membrane channels open up and sodium ions move into the cell. Sodium is positive, and so the inside of the cell becomes more positive. Once this happens, once we have depolarization at one node, that positive charge kind of spreads out, and the whole region underneath this Schwann cell is depolarized. That depolarization is going to affect the next node by opening up all of the ion channels, all of the sodium ion channels at this spot so that more sodium can rush in. So it's kind of like a chain of dominoes. Depolarization and rushing in of sodium here triggers depolarization and rushing in of sodium here, which is going to happen all the way down the axon, the length of the axon, until we get to the very end. I just want to briefly walk you through a little quick video on YouTube, and you can see the link here. It doesn't have any words, but I'm going to just sort of narrate for you. So here we have an outer membrane of a neuron, And we can see that there are some ion channels in the membrane. There's also a sodium-potassium pump. In this case, the sodium is green and there's more of it outside. The potassium is purple, more of it inside. And if we were to put charges in this picture, we would have a more negative inside and a more positive outside. This is when the cell is at rest, and we call that charge difference resting potential. So the charge difference is just set up and is maintained when the cell is not firing an action potential. All right, so I'm going to just hit play. The next phase is going to be depolarization. Notice the sodium channel opened and sodium rushes into the cell. That causes the inside of the cell to become positive. Remember, sodium ions are positive, they're Na+, and so when they rush in, this negative area becomes positive. And that is known as depolarization. We're evening out the charge. It was much more negative in here, and now it's becoming closer to sort of equal across the membrane. The next phase, we see the potassium channels open, and that's called repolarization. The potassium leaves the cell, and potassium is positive too. So as all of these potassiums leave, we're going to end up with a, I went too far. We're going to end up with a return to a negative charge inside the cell. So repolarization, we've reestablished this charge difference. The inside is back to being negative. If you notice though, even though we've repolarized the cell and the charge is back to normal, we have our ions in the wrong place. Sodium is now on the inside, potassium is now on the outside. And before the cell can fire again, that's got to get returned to its original location. So we have this phase here called return to resting potential. And that's going to be where the sodium-potassium pump comes into play. It's going to simultaneously push sodium out and potassium into the cell until everything's sort of reestablished back to its original location. Okay, this video is getting pretty long. I'm going to stop it here, and we will continue the next phase of the neurons sending signals in a second segment. It's almost over, maybe about 10 more minutes.